All right, next we'll look at classification of proteins. Okay, the main message from the section we just completed is that it's a lot easier to synthesize than it is to analyze a polypeptide, but both are possible. Okay, so classification of proteins. Simple proteins will hydrolyze to amino acids only. Conjugated proteins are those that are bonded to a non-protein group, such as a sugar. Those are called glycoproteins. Lipoproteins are when they're bonded to a lipid. Fibrous proteins are long, stringy filaments, insoluble in water, and their primary function is as a structure. Globular proteins are folded into a spherical shape. These function as enzymes, hormones, or transport proteins. Okay, the four levels of protein structure are first primary, which is the sequence of amino acids in the chain and the disulfide links. Secondary structure is that which is formed by hydrogen bonding, and there's two main types of secondary structure. These are the alpha helix and the pleated sheet. Tertiary structure is the three-dimensional folding. Quaternary structure is when we have two or more peptide chains, and quaternary structure is primarily the packing of the three-dimensional packing of these different folded chains. Right, here is the alpha helix. So each carbonyl oxygen can hydrogen bond with an NH hydrogen on the next turn of the coil. Okay, and the next turn of the coil is typically 4 amino acid residues ahead. So it forms this shape like a coiled spring. Okay, in order to form the alpha helix, the amino acids all have to be in the L configuration. Okay, the other type of secondary structure is what are called beta-pleated sheets. And here we have several chains that are hydrogen bonded to each other to form these pleated sheets. The alpha helix is what is present in hair, for example. Hair can stretch, and when it's released, it relaxes back to its original shape. Silk contains only beta sheets, and silk does not stretch like hair does. Tertiary structure is the three-dimensional folding that takes place. And for a typical globular protein, there's segments of alpha helix, and then there's also some beta sheets within that structure. Okay, for enzymes, there is a very specific shape that different pockets need to have in order to function. Okay, so here's an example of primary structure. Okay, secondary structure is the alpha helix and the beta sheets. Tertiary structure, and then quaternary structure. Here we've got 4 chains that are packed in a 3-dimensional arrangement. Okay, denaturation is defined as the disruption of normal structure of a protein such that it loses biological activity. Okay, we don't have to necessarily completely break down or cleave the protein into its individual amino acids. Sometimes disruption of quaternary or tertiary structure is all that you need to denature a protein. Okay, some of the ways that proteins can be denatured include heat or changes in pH. Changes in pH can affect the ability to form hydrogen bonds, for example. If this oxygen, for example, becomes protonated, it can no longer hydrogen bond. Extreme changes in pH would result in breaking the peptide bonds. Heating increases the molecular motion, and it can cause a tertiary structure, for example, to unfold. Denaturation is often irreversible. For example, a cooked egg cannot be uncooked. Okay, what's happening here is you have globular soluble proteins, which when you heat them, okay, the 3-dimensional structure unfolds, and then you end up with some hydrophobic groups on the outside. And some of the other methods of denaturing proteins include heavy metals, okay, such as mercury or lead or silver. Okay, these can cause proteins to precipitate. Mainly what happens here is that the heavy metal will bind to thiol groups, for example. And then alcohol or other polar solvents can disrupt hydrogen bonding by forming new hydrogen bonds of their own.